Thank you everyone for being here today. This is our 43 kitchen table talk. So very excited about that. Welcome to everyone joining us from across the globe. My name is Diana Mitra, your host, and I'm joining you from Viewpoint's Nucleolus conference room here in sunny Boston. As a reminder for those of you who usually come to this talk and a notice for the ones who are first timers, this talk is a live event and it will be recorded. Feel free to turn on your cameras and follow along, but please no on-camera shenanigans. We would prefer to keep the questions to the end of the talk, so feel free to type your question in the chat at any time. During the Q&A, I will call your name, ask you to unmute your microphone, and at that point you can ask your question directly. If for whatever reason you cannot turn on your microphone, let me know and I'll ask the question for you. The event will be posted on condensate.com within a week. So without further ado, it is my great pleasure to introduce today's speaker, Josh Ryback. Josh earned his BA in biophysics with honors from Johns Hopkins University and his PhD in biophysical sciences from the University of Chicago under the guidance of Alan Drummond and Tobin Sosnick, where he made his foray into biomolecular condensates field with a cell paper identifying stress-triggered phase separation as an adaptive, evolutionarily tuned response in yeast. He continued shaping our understanding of the molecular and biophysical mechanisms, understanding underlying condensate regulation in live cells, as a postdoctoral fellow with Cliff Brangwyn at Princeton University. And actually, that is the time when I met Josh during his postdoc, when we collaborated on a paper describing how nucleolar composition adapts the changes in protein expression and the thermodynamics behind this adaptation. So it is really fitting that we're in the nucleolus room today. Josh started his independent investigator career in 2021 when he joined Baylor College of Medicine as an assistant professor. Throughout his career, Josh's impact has been recognized with an impressive number of awards and distinctions, including a Leukemia Research Foundation New Investigator Award, a Searle Scholar Award, and a APSUO Rosalind Franklin Young Investigator Award. Today, Josh will talk about his exciting discovery and characterization of coordinating bodies, which are a new class of disease-driving condensates. But I won't steal your thunder, so Josh, take it away.
Perfect. And thank you so much for that introduction, Diana, and thanks for the whole Viewpoint kitchen table talk team for this unusually, but hopefully I'm not going to jinx it, smooth setup and for the invite. I'm excited to tell you today about my lab's new discovery of this sort of new kind of condensate that results from multiple leukemia mutations and also how some of the rules has impacted our understanding of condensates. And so just to start, I thought I'd first do sort of an overlay of the talk. I think there was also an option to do questions throughout, so I included it, but it could also be at the end, depending. And so for time, I'll just jump into the introduction where I'm going to start with sort of my lab's broad interests and then funnel into the key parts that led to the story that I'm going to tell you about today. And so my lab, like the room, I guess, that Diana's in, is really excited about the nucleolus. And so the nucleolus is the location of ribosome biogenesis, but in the cell, here's how it actually looks. And so on the top left, you can see a picture of a nucleus where the most abundant nucleolar protein that you'll all know way too much about by the end is endogenously fluorescently tagged with Ed and Sherry. And then it's also shown in the fire mode, which really for condensates lets one really see the dynamic range of localization patterns. And then the scale bar is 2 microns. And so my lab's really excited about how compartments like the nucleolus do their functions. And of course, the nucleolus, as I'm sure all who sign up for this are familiar with, isn't the only condensate. And even within the nucleus, looking at sort of chromatin, you can easily see how many other condensates are sort of squeezing their way in between histones and pushing the chromatin away. Now, of course, not all condensates are not pushing directly on chromatin, and the one that's going to be most relevant for the talk today is going to be these aberrant condensates, especially the ones that people have seen with nucleoporins, and that seem to recruit core regulators and drive cancer. And of course, what's really the excitement behind this, and differs from sort of the nuclear body field, is this notion that even though these systems are really complicated in the cell, that they can form sort of like oil and water, but with much higher complexity. And so really, to summarize, my lab is really excited about that concept and how we really shape that into the rich biochemical functions that condensates have, both in normal physiology, but also in disease. And so, of course, a lot of what we've learned has been from reconstituting simplified condensates in the test tube, where we've learned about things like weak interactions between intrinsically sorted regions and various substrate binding domains, and then also sort of the extension of multivalency and network interactions like the PRM and the SH3 multivalent system. And of course, a critical part of this is the essence of phase separation, which is the idea that you can simplify your system in terms of a phase boundary or a phase diagram. And at least for me and for my lab, a really critical part of this is this notion that, unlike the membrane-bound organelles that really have sort of a strict boundary, condensates allow for steady-state partitioning, and that can be a really good proxy for learning about the thermodynamics and what's driving things to the condensate or the condensate to form itself. But while reconstituted systems have taught us a lot, and have really been great for these basic principles, their simplicity really makes mapping them to sort of complicated biochemical processes that happen within cells really difficult. And of course, on the other side, for condensates in cells, their sort of complexity that really holds that information also makes them difficult to perform measurements with the same quantitative rigor and depth of understanding. And really what drives my lab's approach is really this idea that complexity, or really the hypothesis that, well, maybe there's additional complexity that may arise in cells and under these more complex systems. And for this crowd, I guess I thought it'd be best to summarize this in 2 ways. And so I think that the emergent biophysics that could exist may yield additional principles of sort of how cells can have new modes of biochemical control, but also in the context of disease may yield insights that sharpen how condensates are changed or how aberrant condensates are formed, and that may provide key foundation for therapeutic insights. And so how do we do this? And so really my lab is really just focused on this concept of sort of pushing the forefront in trying to understand condensate principles by directly measuring everything in cells. So we have a very well calibrated microscope. I actually just aligned it yesterday after a year because we take daily power meter measurements. So it's sort of like our physical chemist calorimeter. And then we turn that into, cellular measurements and then interplay with sort of quantitative microscopy, mostly inspired from physical chemistry concepts. And then, of course, through perturbations and mutations, or using, uh, you know, uh, therapeutic agents such as drugs, we try to build models and really try to gain some mechanistic or therapeutic insights. And so really the core elements of my lab are really this notion of building new tools and new models to understand condensate form and function of physiology and disease. And so today I'm going to talk about, in particular, one new model that really ended up being much bigger than we initially thought it would be. And that was really spearheaded by a student, Gandhar, who is a recently graduated MD PhD student. Actually, he recently started his clinical started as his residency at Harvard MGB. And he was a jointly mentor between my lab and Peggy Goodale's lab. And additionally, key to this project was Elmira, a postdoc in Peggy's lab. And so now that I've sort of told you about my biophysical interests, I'm going to briefly mention the background for the focus of what's going to become this new condensate, which is hematopoiesis and acute myeloid leukemia. And so hematopoiesis is basically the generation of new blood from blood marrow. And basically, blood stem cells undergo a standard process in the marrow into our blood, where the stem cells go from progenitor cells to precursor cells, and finally myeloid and lymphoid cells. Now, in the context of acute myeloid leukemia, that doesn't quite work as well, and that's because certain cells, due to mutations, get sort of stuck in the progenitor-like phase. And in the case of acute myeloid leukemia, they get stuck in this myeloid leukemic state. And so AML is particularly a pretty big clinical burden. It's about 20,000 new cases per year in the US, and it doesn't have a really great 5-year survival rate. And so that makes it of interest. And additionally, from the sort of basic cancer biology side, the AML or a particular class of AMLs are sometimes called high HOXA9 AMLs, have really focused on a few set of leukemia mutations that are very different, but seem to act effectively similar in terms of the downstream cellular and therapeutic properties. And so notably, it's NPM1, so this nucleolar protein mutations, along with nucleoporin, here the R is rearranged, or nucleoporin oncofusions, and also KMT2A, where some of you may be familiar with it as MLL rearrangements or oncofusions. And so what's really surprising about these is that they seem to really funnel to activate a very common gene profile that sort of keeps them in this leukemic state. And additionally, from the clinical side, 2 sets of inhibitors have really been successful for will have shown some promise in all of these contexts. And so there's this broader idea that these few things may relate to each other. And so initially, we sort of had this idea like, well, maybe there's some common mechanism of action. And, in case you forgot where you were, spoiler alert, condensates are going to be involved, and that's where these C bodies are going to come into play. And of course, I tried to warn you up front, you're going to be knowing too much about this NPM1 protein, and that's where the sort of story is going to start. And so NPM1 has this sort of basic structure, very common to condensate forming proteins. It has an oligomerization domain that allows it to form a self pentamer, an intrinsically disordered region, and a RNA binding domain. And good both for its ability to phase separate, but also in terms of imaging and microscopy. It's one of the highest proteins in really the majority of cells, so about top 10 in most cancer cells and top 50 in most cells in general, or I'll say most human cells. Additionally, in the context of adult AML, it's not just some minor rare mutation. It's really it's really one of the top, if not the top, most common driver mutations in adult AML. And it really results from a small frame shift that really changes very little of the amino acid sequence. And actually, initially, it was found so easily because NPM1 itself is used as a histological marker in cancer pathology to assess aggressiveness. And so when stained, it actually stains the cytoplasm. And so it's historically had this name, cytoplasmic NPM1. Additionally, later it was found out that that's because this frame shift causes a de novo site to bind the major exportant XPO1 that can export it into the cytoplasm. However, recent later results really seem to suggest that it somehow was very directly regulating these leukemic genes, and it really was a confusing result how the cytoplasmic protein could do that. And then of late, studies while we were really investigating started coming out with this notion, well, maybe it is in the nucleus. And so that's sort of the backdrop to the story, and really yields the question of what's going on, what's happening here. And so just to summarize the first part, the introduction, I sort of briefly told you about my lab's interest to sort of find out how condensates work in more complicated systems. This idea that, well, what's going on with these distinct, AML drivers that are so different but seem to act very similarly, and then some depth on NPM1C, which is a very unclear case of how this is working. And so now we'll get into the crux of it, which is really the backdrop of this C body. And so basically, as my lab is really focused on really trying to create new models and using quantitative microscopy, we rely heavily on patient-derived cell models and related patient-derived systems to really do our work. And so the first steps was really to take some of these cell lines that were from patients who had NPM1 mutant AML and also are still dependent on NPM1C for growth and other features that we'll discuss later on. But here you can see is an endogenously tagged live cell with NPM1 wild type labeled, and you see what you expect, which is NPM1 is in this sort of ugly smush chocolate chip cookie region. And then it's also found, albeit less strongly, in the nucleoplasm labeled with N. And so now if we compare it to in the same cell where NPM1C is also in endogenously edited with a fluorescent tag, we see what would be expected. It's in the cytoplasm, and also what we could expect given its binding to XPO1, which is it seems to have some enrichment on the periphery, suggesting that it's at the nuclear pore slightly more than the cytoplasm. But unexpectedly, we additionally find that it's in these bright nuclear puncta and really not showing any preferential enrichment in the nucleolus. And this isn't just this one cell, but it's in, throughout the population. It even more striking, these small puncta seem to actually enrich NPM1C better than NPM1 wild type is enriched in the nucleolus. And so that was a really surprise. And before proceeding, we also wanted to make sure that this wasn't just a fluke of the cell line. Cell lines are pretty far from actual patients. And so we tested other cell lines, patient-derived xenografts models, and even antibody stained with cutting-edge anti-NPM1 antibodies in near-direct patient cells. And throughout all this, we see the same consistent features. And so what's going on? How does this spatial separation occur? And so first, why am I really asking that? And so some background, of course, it's cancer. And so a key part of that is you need to divide pretty frequently. And a key part of division is actually breaking down most of your compartments, including essentially all condensates. And for NPM1, and Diana did actually a lot of work on this. NPM1 actually becomes phosphorylated, and that results in it not being a pentamer, and it contributes to nucleolar disassembly, where it goes for monomers, and then after cytokinesis, and then you have two daughter cells, then it gets dephosphorylated, reforms pentamers, and helps the nucleolus reform. Now, of course, in the context of NPM1C and NPM1 mutant AML, this creates a little bit of a seeming problem based on our initial results, and that's that NPM1C and NPM1 wild type seem very separated into distinct pentamers, one seemingly in these foci or condensates, or eventually we'll call C bodies, and then NPM1 in the nucleolus. And so when they both get phosphorylated, because again, NPM1C has 95% of the sequence, and so those sites have not changed. They're on the whole other side of the protein. Then the question becomes is how do they get back together? And the reason, of course, why that seems a little non-intuitive is because you'd expect that they should mostly just randomly mix, because the oligomerization or pentamerization domains are exactly the same between the two proteins. And so you can imagine something like somehow there's some oligomer sorting that happens over time, or maybe they actually phase separate pre or during that pentamerization process. And so that's really where we started, and really to try to gain some insights into what's going on and how this happens. And so I have a cool video of this. I hope people can see it okay on their screens. But here you can see NPM1 and NPM1C go through mitosis. So here's metaphase. Every second here is about an hour. And then you can see rapidly when they undergo cytokinesis, they separate and form condensates. And so as that was really fast, I'll quickly walk it through. And so as expected, mitosis, resets NPM1C and NPM1 wild type and removes them from condensates, and then largely they're diffuse throughout and during metaphase. However, during anaphase and telophase. They both enrich on what's called the chromosomal sheath. And so basically they're at or on or essentially just adjacent to chromatin. You can see that sort of here. And NPM1C is also there, albeit a little bit less. And then rapidly, they seem to go to very distinct places where NPM1 is starting to form what are called pre-nucleolar bodies, and maybe we'll call it pre-C bodies, but very distinct locations. And then additionally, what was also apparent is that in early G1, a lot of NPM1 wild type seemed to be still in the cytoplasm. And so from doing this on a bunch of cells and with really fast or as fast imaging without toxicity, we could do some quantification. And so one, you can see sort of a summary in this plot that shows NPM1C enrichment with NPM1 wild type and shows sort of that progressive enrichment together and then rapid within nearly 3-minute separation or demixing that would be consistent with it separating during the oligomerization process. Additionally, once the periphery is established, NPM1C and NPM1 wild type really have different sort of allowances per se to go into the nucleus, where NPM1C sort of really has no change in its nucleoplasmic and cytoplasmic levels, whereas NPM1 wild type slowly enters the nucleolus, potentially more consistent with a slow second stage of self-sorting that occurs. And note, this is really different than what you see in other cell types. For example, here's U2S, where this normally happens on the order of 15 or 10 minutes, not 3 hours. And for those of you who could be skeptical, like one of our reviewers was, when we overexpress NPM1C in U2OS, it actually does recapitulate this delay, shifting it, and note the difference in time scale. And so now that we really established what these are, that these form and they seem to really happen rapidly and sort of consistent with the signs you would expect for a phase-separated condensate, we wanted to take a step back and ask, well, how do we distinguish exactly what's here to basically understand more about how it works, but also to know if it forms independent of the NPM1C. And so initially, we sort of had a hunch that it would relate to those chromatin regulating proteins that are targeted. And so in particular, we focused on XPO1, NUP98, and KMT2A menin because they're sort of the primary targets. And so here you can see that when we fix these cells and immunostain for those markers, in each case, those proteins are also in bright, are also contained within or enriching within those condensates. And here you can see sort of a radial enrichment from the center of the NPM1C signal outwards for the other proteins. And so then we thought, well, if this is a chromatin regulated component, maybe it's also involved in activating the chromatin. And consistently we see polymerase 2 signal there, active marks, and of course, just as a negative control for what this would look like, we've also ruled out a bunch of your sort of standard other nuclear bodies or nuclear condensates. Finally, we've additionally confirmed that not only are those proteins there, but it's also found on the HOX genes itself. And so it seems to be that the C body is directly activating, or at least that's our model. And this is sort of consistent, or we were able to show that these 4 proteins are also co-enriching with sort of your more standard chromatin localization sequencing assays like cut and run. Okay, and so now that we know some things that are there and have gained some insight, we now are really able to ask, is NPM1C actually necessary for any of this that's going on? And so, of course, what we utilize is our Peggy's lab prior to this had published a inducible degradation model of NPM1C, where when you add this molecule called DTAC13, it induces acute degradation on the order of, let's say, 3 to 6 hours. You get about 80% degradation as shown here. And then now we can ask what happens to those key leukemic regulator proteins. And so here's one of them, the one that interacts with NPM1C, XPO1. You can see whereas in the control, there are these bright co-localized foci, those actually go away. And this is not just true for this one cell, but and for XPO1, but it's really true for every cell and for these 4 regulators. And because of that, we started calling these sort of coordinating bodies, with the notion that it's sort of coordinating the co-focal enrichment of these proteins. And so from now on, because it's been hard, I'm just going to call them C bodies. And so this, of course, shows necessary, but is it sufficient? And so, of course, that's the notion there, is that right phase separation is this thermodynamic minima, where you minimize your system and separate into 2 phases, a dilute and a dense. And so the question is can we now add back and then degrade and see if there's a rough concentration boundary? And in cells, especially degron models, where not every cell is degrading at the same level, it's a little tricky. But in general, we did see signs where at the lowest cells, we did not see condensates and so here's fraction of cells where one plane is showing a identifiable condensate above a threshold, and that seems to get bigger as you overexpress. Additionally, because it's difficult, we also went to other genetic systems that don't have NPM1C to ask the reverse: can we induce NPM1C formation? And we found it was actually quite independent of genetic context. And so here's a common suspension line, HL60, human CD34 blood cells with NPM1C overexpressed. Additionally, in all these cases, including in UTOS, but here you don't have the 4 standard leukemic markers showing strong focal enrichment. But when you add an NPM1C, now even in these unrelated to NPM1 mutant contexts, you now get them being enriched together and consistent with a phase boundary. And so from here, I'd like to close this section. And then I think I'll stick with, unless, Diana, would you prefer I just wait till the end?
It's up to you. I actually have a couple of questions. Is anyone else in the room?
Perfect. I think we have time for a minute or 2 of questions.
Yeah, so I was curious about it was really cool that the pentamers kind of sort themselves out. I was wondering if you went deeper into the mechanism and whether binding to RNA has something to do with it, given that the NPM1C has a RNA binding domain that's kind of defective of binding nucleic acids.
Yeah, so I think that's a great question. So we have done a little bit of follow-up on it, actually both anecdotally in my lab, but also things I've seen before my lab. And so even if you're a standard protein, let's say just delta RBD, you actually still will go to the chromosomal sheath and actually behave just a little bit weaker, like NPM1 wild type. And so it doesn't seem, at least, that it's rRNA dependent, if that's sort of what you were meaning by RNA. In the context of the C body, we don't yet know if RNA is critical there or not for its formation.
Got it. Thank you. All right, and I'll ask you the other question after the next session.
Okay, perfect. Well, if there's nothing else, then I'll just continue for now. Okay, and so now that we've really established that NPM1C is necessary and sufficient, really the question is how do these C bodies work? What do they depend on? And really, how is this 5 amino acid or 7 amino acids, 5% rough difference between the two proteins causing such a striking difference in protein phase separation? And so notably, for context, sorry, I skipped over that. But anyway, this is the third section. Notably, of course, while condensates were initially, discussed in analogy to oil and water, of course, proteins and nucleic acids are much more complicated than oil. And so therefore, they're never exactly perfectly fitting into that. And so, of course, the simplest case is that there's many components in the cell. And so then, so you're going to depend on the ratio of those two components, and they're favorable to interact together, which is usually called in the field heterotypic interactions, or with each other homotypic interactions. And so previously in Cliff's lab, we developed this assay where basically it allowed you in cells to overexpress one protein and then ask how the partitioning or the related free energy of transfer changed as you overexpress that protein. So despite the nucleolus getting much, much bigger, NPM1 in this case doesn't actually like the nucleolus as much despite that size difference. And so that roughly is able to quantify the heterotypic interactions. Additionally, I'll briefly mention there's even more complexities emerging in the literature with this notion that really networks and of interactions and inhomogeneity and some other exciting things from multiple groups. And it sort of creates behavior at small scales that maybe we'll briefly touch upon, but largely I'm going to stick with introducing this idea of heterotypic destabilization. And so largely, first we want to know is it, what is driving these C bodies? Is it just predominantly NPM1C interactions with itself, or is there really a role for heterotypic interactions? And so a way we thought to do this, and also for those of you who are more maybe in the clinical direction of relevance to agents that are used to some extent, depending on which one in the clinic, we focused on those. And so those are, as I mentioned previously, are XPO1 inhibitors, here shown in blue. This is Altanexor, which is one of the ones available, and VTP, which is a menin inhibitor, which binds the interface between KMT2A and its partner menin, but called menin inhibitors. So if one adds Altanexor, it's well known that then sigh of relief, it actually isn't just messed up in some unknown way, but it actually will go into the nucleolus, which again you can see by maybe not as bright, but still smushed chocolate chip cookie. And so, of course, under those conditions, when that XPO1 NPM1C interaction is broken, now we also additionally again get that the core chromatin regulators we looked at don't go too bright don't go to bright puncta. So seemingly that interaction is essential for C-body formation. Additionally, we applied VTP or menin inhibitors, and in that case, we did not see noticeable loss in condensates. However, when we looked at the individual proteins and their recruitment, all were still recruited to C-bodies except for menin. So it seemed basically like the menin inhibitor was able to push, per se, menin off the C-body, or phrase maybe another way closer to what's mechanistically going on, that menin was only in the condensate, presumably due to that interaction, and thus compositionally changing it. And so this really shows at least one heterotypic interaction, that between XPO1 and NP1C. That's important. Additionally, I'll just summarize that we do not just analyze n equals 1 pretty cell, but do this on dozens of cells to really show that it happens at a population scale. But for time, I'm just going to gloss over that and refer you to the paper, and hopefully you'll trust me. If not, you're free to read up more there. And so now that we've established it's heterotypic, we thought, well, maybe we can go back to that idea of heterotypic destabilization and try to really tease apart or begin to open up how important C-bodies are for things like cell growth and things like that. And so first, we overexpressed NPM1C without degrading the endogenous, similar to what I showed before with NPM1 wild type in different contexts. And you can see the condensates get really big, but also does the localization in the cytoplasm. And so if we quantify that with overexpression level, what we see is that actually the C-body becoming destabilized doesn't actually, while it doesn't actually strongly increase nucleoplasmic levels, it actually allows for better export of the NPM1C protein, a sort of indicator of a weaker C-body formation. And not only that, but heterotypic destabilization also results in selection. And so the cells actually grow slower that are expressing high levels of NP1C showed here. So they actually get selected out of the population, which, of course, makes them a little bit more difficult to study. And anecdotally, we've been told that others have had difficulties studying NP1C for that reason in the endogenous contexts. And so with this in hand, and that heterotypic destabilization seems to be a feature, we thought, well, maybe we could push this and sort of ask, well, which interactions and how do they work together? Which features, which sequence features of NPM1C actually drive C-body formation? And so to do that, inspired by studies and domain assignments, mostly from Richard Wacky's lab that Diana was a part of, we did a full truncation screen of all the regions in the IDR and the C-terminal domain that are classically annotated. And so the idea basically is that when expressed at low levels or extrapolated to low levels, we could look at the enrichment or the amount in the C-body versus in the nucleoplasm as a measure of the stability and the role of that of that domain that's being deleted in the interaction. And the notion broadly in the field is this idea of sort of weak interaction spread along the sequence of a protein, sort of what I'm going to call non-cooperative interactions. And when we look at the data, we were really shocked to see something really much really not consistent with that. And in fact, most, if not all but two of those truncations result in a more than 10-fold decrease in their ability to enrich in C bodies. And that's more consistent with a cooperative network of interactions, not sort of weak spread across, but really all working together. And so from here, we can also do the other assays that I showed on the overexpressing the full length. And so again, only those that still partition well are the ones that really hurt viability and result in the best, the strongest response of the endogenous to be pushed into the cytoplasm. So strong signs of heterotypic destabilization. Okay, and so of note, you can see that if you focus on the three that really were those outliers from the tenfold impact in partitioning. And the reason why I'm highlighting them is because now we then asked what happens when we actually degrade the endogenous NPM1C, which ones can rescue, so are able to form condensates. And so here's the percentage of cells that I showed previously versus overexpression level. And again, only three, including the sort of wild type full length, are able to form condensates. Additionally, here I'm showing differentiation marker after a week in culture, showing that only those that are able to form condensates are able to prevent or maintain the leukemic state. So that continue to grow. And you can see that here where I've highlighted that only those that are able to form condensates and were those few that were partitioning highly are still now the only ones that are able to grow. And so from here, I'm going to quickly switch. And although it may not look like it, I promise, Diana, I'm good on time. And so basically from here, Peggy's lab really we took one mutant in particular that seemed to be able to still be in the nucleus and a little bit in the nucleolus, and additionally was still in the cytoplasm, but was not able to localize to C bodies to use as a strong tool to ask about the role of C bodies in maintaining various leukemic features. And so we compared overexpressing those just in OCIML3 after degradation and looked transcript wide and saw that only the construct that formed C bodies was able to maintain the leukemic transformation. Additionally, we looked at the ability to stall differentiation when switched to an appropriate media that is supposed to induce the differentiation. Only those with C bodies was able to delay that substantially. And finally, Peggy's lab actually engrafted and showed the ability for in vivo expansion of these constructs in mouse. And again, only C bodies seemed to be necessary broadly in these assays for maintaining the leukemic state. And so from there, and I think I'm going to quickly just skim through the next section, which I promise is on the shorter side, Diana. But here you can see really we see the fundamentals of how the C bodies worked, and also that allowed us to look at their role in maintaining the leukemic state in these assays. And so finally, this idea of disparate mutations converging on C bodies is really begins with this notion that Rick Young and Richard Kawaki's labs have been, along with others, have been pushing, which is this idea of many labs of fusion oncoproteins and also other pathogenic variants being prone to condensate formation. And really there's a wide depth there, and I think there's a lot of interest in that space. And I sort of want to phrase it to preface maybe a little bit bigger, where we're going to go at these in the narrow AML field of this sort of broader idea of how do we think about that? It almost seems like a daunting idea that there could be so many different ways or so many different types of oncogenic condensates. on one hand, or maybe there's some way to converge them or bridge them together. And so again, could that be the C body for our nucleoporin oncofusions and these KMT2A or MLL rearrangements? And so first we asked, in our hands, did they in U2OS when overexpressed independently? So no NPM1C here, and they're each expressed alone. Do they form bright puncta? And they did, and I'll show it at the end that they seem to be consistent with having a CSAT. Additionally, we were able to see that the same core proteins were being recruited, XPO1, menin, KMT2A, NUP98, to these de novo condensates. And here we were a little bit initially like, well, maybe that's good enough. But of course, they could be acting either as more like clients or more like scaffolds. It doesn't necessarily tell us if they're actually forming via the same mechanism. And so we thought about this a little bit in a different way. And so I'm going to skip that slide for time, but the way we were thinking about this is if the proteins sort of are, imagine NUP98 is just FG repeats and NPM1C is more unfolded RBD interactions, and they actually don't really need to interact that well, but can actually drive phase separation independently or distinguishably, then you could, with high overexpression in the same cell of NPM1C or of an oncofusion, form distinct condensates that would both recruit the downstream leukemic proteins, but actually form condensates in a distinguishable, biophysically distinguishable way. Or on the other hand, they could be indistinguishable. So they could actually be sort of enhancing similar interaction networks. And therefore, one would never get different condensates within the same cell and only have compositionally dependent condensates depending on the ratio between the two drivers. And so, long story short, we have yet to see a case where they were able to form distinguishable condensates. And so here's just an example of what that looks like, where there's just complete overlap. And when one is a little bit dimmer due to being slightly out of Z, so is the other. And so it really seemed to suggest a shared scaffolding network and maybe a potential possible targetable entity. And so that sort of ends my final part, showing that these other oncofusions can form puncta that recruit C-body associated leukemic regulators, that they are biophysically indistinguishable and therefore seem to be, from NPM1C driven C-bodies, and that together these results really suggest that C-bodies may be a unifying feature and therapeutic vulnerability across multiple leukemia subtypes. And from that, we're done. And so I'll just end with a little quick summary. One is I think, hopefully I've convinced you that C-bodies are a little bit unexpected, especially in the ability for NPM1C to form them separate from NPM1 wild type, and sort of the unexpected cooperativity amongst its sequence for doing that. Additionally, just to summarize maybe a little bit more of the big picture cancer/therapeutic elements, we discovered this C-body, demonstrated that it seems to be essential for cell growth, differentiation, and expansion, and then have proposed it as an emergent phenomenon that may drive these distinct leukemia subtypes. And from there, I want to conclude and thank everyone involved and everyone in my lab, especially Gandar, who, if you're in the Boston area, is going to soon be looking for postdocs and potentially industry during his residency. And so I highly recommend him, and definitely reach out. And I hope you all enjoyed, and I'm really excited to answer any questions.
Thank you, Josh. Are there any questions online? Anyone in the room? Yeah, John has a question here in the room.
Hi, Josh. Thanks for the great talk. I was wondering are there any known kind of therapeutics out there that are known to target one subtype of AML that you could test to see if they kind of unexpectedly also target these other subtypes, given the multiple factors coming together in the same condensate to kind of support the therapeutic angle?
Well, so a little bit, we focused on menin inhibitors so strongly because this was sort of an emerging avenue that was unclear at first if the menin inhibitors, which were more developed for the MLL or KMT2A rearrangement subtypes, would work. But beyond that, we've done a little bit, but not as extensively. That's a great question.
So you mentioned, just following up on John's question, you showed that beautiful data with the XBO inhibitor that it dissolved the NPM1C foreign C bodies. Do you see the same thing with other fusions? I mean, do XBO inhibitors dissolve all C bodies, regardless of what fusion they're originate from?
Yeah, so we have some new results on this. We didn't go into we went into it a little bit in the paper, a little bit more in the context of the menin inhibitors, which did act this way. And it does seem like XPO1 inhibitors in some of them will result in dissolution.
Well, so do you think that XPO1 is one of the steady scaffolding proteins then?
Yeah, definitely. Right, and I think it really highlights, that NPM1C seems to really be hijacking it from sort of that network of interactions, right? And really gives this picture of sort of this shared underlying network that something like oligomerization and causing phase separation may be sort of hyperactivating when cells are trying to actually turn it off. And differentiate is sort of the idea.
So have you looked at all, done like a broad, I guess bioinformatics or data science approach to kind of figure out which fusions might contribute to C bodies, and do all the proteins that you've seen so far that are driving C bodies, do they have strong XPO binding sites?
We've done this a little bit as far, these NES predictors have strengths and weaknesses. And so it was largely not very clear. Notably, even in the context of sort of even NUP98, right? It's not clear that there's a strong, that would even come up as a strong signal in that sort of an assay. Or bioinformatic approach, I guess I should say.
Yeah. Well, that's fascinating. Join me in giving Josh one more round of applause. Thank you everyone who joined us today online and in person. And thank you to the team that worked behind the scenes to make KTT happen. So thank you to Jennifer Tallman for editing and marketing, and Bobby Ingram for IT support. As I said at the beginning, the recording will be posted on condensates.com and YouTube within a week. Take care. Have a wonderful summer and see you at the next one. Thanks so much, Josh.